So we're going to talk about, this is the procedure here for proper virus isolation. I'll use my laser pointer. So we have a sick person here, right, with a runny nose. And as Tom always says, we're going to take the snot and put it in a test tube here, okay?
Scientific term.
And then you're going to take that snot, right? Now remember, this snot is something, it's stuff your body's trying to get rid of because you're sick.
No, it's not.
Right? So it has all the junk and bad stuff your body doesn't want in it. And, you know, the purpose of this experiment is someone hypothesizes that there's a viral particle in there that caused that illness, and we're going to find it. And this is the way you would go about finding it. So once you have it in the test tube, you put it through a filter, and that separates things by size. So things that are big, like bacterial cells or white blood cells, will not pass through. So it's just a way of separating out things that are really tiny, okay, but there's still a lot of gunk in there. And then you're going to put that through, this is a density gradient ultracentrifugation. Really fancy sounding word, but all they do is as you can see in the top left test tube, they have sucrose at different concentrations. So it's less concentrated at the top, more concentrated as it goes down. And what those different concentrations do is they have a different density, right? Which is the amount of mass per volume. And so that when you put another material in there and spin it around really fast, right, causing things to go toward the bottom of the test tube because it's sticking out, like you've all been on those rides at the amusement park where the floor drops out and you stick to the side wall when it's spinning around. That's a human centrifuge. Okay? So this one goes a little faster, but it does the same thing. And so that material gets pushed down toward the bottom, just like you get pushed out against the wall. But it kind of stops when it meets the area of sucrose that's at the same density as that material. So things end up separating by density, and you can see that there's this dotted line here. Well, that represents the band where the envelope membrane of the virus, okay, which is the outer coating of it, would settle after spinning it in the procedure. And then you can stick a syringe or a pipette right into there and just remove that band. Okay, so this is the purification method. This is how you separate all the particles from each other by density and from any other materials that are in there. And as you can see, when you look at this under the electron microscope, you see particles that are identical to each other in shape and form and size. And these are actually bacteriophages. And bacteriophages are these microscopic nanoscale particles that can be reliably shown through this procedure over and over again from bacterial cultures. They were originally thought to be viruses of bacteria or compared that way, but subsequent research actually showed they have a beneficial survival benefit and only really come into existence under extraordinary difficult situations in the laboratory. But nonetheless, since these things have been purified, you see that under the microscope, that's the only thing you see. And they're easy to identify because they have this distinctive hexagonal shape with a tail. And there are different phages that have been found in different types of bacterial cultures that show different distinct shapes that are similar to this. And you probably have seen many of them. Some of them look kind of like space aliens. So this is the basic procedure. It's quite straightforward. And then you have to do, of course, at least a negative control experiment to make sure that what you have is only associated with a sick person, but not with a healthy person. So here we have this super healthy doctor, right? That doesn't really exist in life. But because, you know, doctors don't live as long, they have higher rates of suicide, drug addiction. So, but nonetheless, so you take his healthy snot, right? Probably he's loaded with vitamin C, and put that through the filter, do the centrifugation, purification, and what do you see? There are no particles, right? It's a blank slide because he's not sick, so there's no virus that allegedly would only be present in sick people. Now this is what you should find, and it's never been done for bacteriophages, obviously, because they've never been proposed to cause disease. But if you were looking for a phantom virus, you would have to do this procedure to make sure that it was only in sick people and not something that was just present in everybody. Common sense. Now this is what the experiment that is actually done. Okay, now they call it virus isolation, but you notice I put that in quotes because they're not using the common definition, they have a special definition, right? So really what this is it's a cell or a tissue culture. So the first part is the same. They start with the sick individual, take the snot in the test tube, and then filter it. But then they don't purify it further. Instead, they add it to a mess, okay? And this mess is a foreign cell culture, and I'm sure you've heard that it's usually monkey kidney cells known as Vero cells, but there are some others occasionally used. And that culture is on a starvation diet with minimal essential growth media, and it's given toxic antibiotics, usually things that are specifically toxic to that type of tissue, like nephrotoxic antibiotics, if they're using kidney cells. And then other biological agents like fetal calf serum that also contain genetic material and other biological material and would be, toxic to monkey cells. Like if you put cow blood products in our body, they would be, you could get an allergic reaction or other types of toxicity anytime you put materials from other species, into your body. Not eating them, because then they're processed by the acid in your stomach, but in a cell culture, there's no digestion. It's just exposed right there to the cells.
Wow.
So essentially you have dead and dying cells, and then you have a picture like this, which, it's hard to see what this picture is. I can tell you what they say it is exactly, but you can tell right away that there's no things that are identical that look like a group that you see everything looks the same, like there's no uniformity, right? There's a lot of different things in here. And what this is it's actually, this is a vesicle inside of the cell. So, and then these particles, they say, are SARS-CoV-2. But how do they know these ones are and these ones aren't? Right? It's just completely arbitrary that they say that. And then the coup de grace is, what about the control experiment?
Right.
Oh, they don't actually do one.
Right.
The reason they don't do a control experiment is that if they did the proper control and took the secretions from a healthy individual, they'd have the same exact results and the same exact microscopic pictures, because they created the recipe for the death of the cell culture, and that's essentially what they're showing in the pictures. So, to summarize, if no virus exists, these are the results. There's no known structure or composition of a virus that doesn't exist. In other words, the spike protein, which is the gene that they put in the genetic vaccine, what is it? Doesn't exist. Okay? Can't know anything about it. It's made up. There's no known genetic sequence. Of course, how could you know the genetic sequence of something you haven't shown to exist? There are no mutations or genetic variants, of course, because there's no original. There's no way to develop a test. What would you base the test on? And there's no way to demonstrate that this imaginary particle causes a disease because it hasn't been shown to actually exist. So this is the basis of where we're at. So why don't we take 5 minutes to run that right now. Now, anyone who wants to talk to me during that time, come on.